My name is Ashley Volz with Antibody Engineering, and today I'm speaking with Pia Abola, Director of Marketing at Applied StemCell, who's here to talk to us about ASC's technology for mammalian cell line engineering.
Thank you, Ashley.
Thanks for being here. And I'd like to start by asking why therapeutic antibody developers should consider changing their workflow to screen and validate directly in mammalian cells.
Well, the answer to that question is time, money, and increasing your chances for success. If we think about why we screen in bacteria and yeast, it's because the technology hasn't been there to achieve the diversity to explore a wide range of conformation space. In bacteria and yeast, you can achieve library sizes of 10 to the ninth to 10 to the 10th. But the molecular environment of bacteria and yeast is very different from mammalian cells, where we want the antibodies to work. So we may miss leads that would have worked in mammalian cells or get false positives that would never have worked. Efficient approaches to insert expression cassettes into mammalian cells, like virus-based mammalian display, are not efficient enough to achieve enough diversity. However, our target technology is efficient enough to achieve the required diversity and includes insertion into a proven safe harbor site for fast pool-based screening.
Now, Target technology sounds very interesting. Can you tell me how it works?
Yes, I really like this technology because it is similar to what I used to work on in graduate school. The target platform is a three-component system that includes a donor plasmid with your gene of interest and a target insertion sequence, uh, a target integrase expression plasmid, and a cell line that has a landing pad integrated at the target safe harbor site. The target integrase is a serine integrase that delivers highly efficient integration of large DNA cargos. We recently demonstrated insertion of a 50 kb sequence at over 85% efficiency. It is highly specific for the landing pad, so we get negligible off-target events, and the safe harbor site enables reliable expression that is consistent from clone to clone. This means you can use the platform for engineering cell lines used in pool-based screening and automated pipelines. And unlike CRISPR, the IP landscape is very simple.
So you mentioned CRISPR. How does Target technology compare to the other DNA insertion platforms such as CRISPR?
Well, if you look at this slide, you can see how target technology compares to CRISPR, lentivirus, and transposon-based insertion methods. Target is the only platform that delivers efficient, large cargo, site-specific, single copy insertion with consistent expression. And as I mentioned in the previous slide, clear IP for commercialization.
That sounds great. Are there any other applications for Target technology?
Yes, it is compatible with inducible expression, so you can express difficult proteins, and the large insertion capability mean you can express genes that assemble into multi-protein complexes while maintaining strict stoichiometry.
Well, that sounds great. Thank you so much for speaking with me today.
Thank you so much for talking with me.